Good morning. My name is Karuniawati. I'm from the Department of Microbiology, Medical Faculty. The learning objective of my lecture today is about molecular pathogenesis of infection. To learn the pathogenesis, of course, we have first to learn host-parasite relationship, and I will focus in basic concept of infection. As we know that the disease will be produced by three main components. They are host, agents, and environment. When the three of them are in balance, then there are no disease. But when one of them or two of them are imbalanced, then the disease will appear. When we talk specifically about infection or infectious disease, we have to understand about Koch's postulate, which is stated by Robert Koch in 1890, and the postulates contain of four important things. One is that the microbe must be present in every case of the disease. The second one, the microbe must be isolated from the disease host and grown in pure culture. Means that we have to prove it that the microbe is there, that the microbe is in the specimen of the patient. The third postulate is the disease must be reproduced when a pure culture is introduced into a non-diseased susceptible host. Means that if we get the isolate from the sick person or from the patient, culture it, purify it, and then introduce it into the healthy person, then the healthy person is becoming sick. The last postulate is that the microbe must be recoverable from an experimentally infected host. It means that the microbe should be able to the last postulate means that we should be able to isolate the microbe from the second infected person. There are some exceptions to Koch's postulates. There are, first, the microbes could not always be grown in the laboratory. For example, some bacteria like Mycobacteria leprae, Chlamydia species, and also viruses could not be grown in the laboratory in vitro. So we have to grow this kind of microbes in the live cells. The second exception is cofactors or genetic and immunologic factors in the host may play a vital part. And I think it is clear that the immune response is very important thing to defend the disease. The third exception is that the ethical consideration prevent applying Koch's postulate to diseases and pathogens that occur in humans only. You remember in the previous slide that to prove that a disease is an infectious disease, we have to introduce the microbe to healthy person, and then the healthy person has to get the same sign and symptom as the first patient. In some instances, this cannot be proven because the ethical consideration. We can prove it, of course, using animals, but not human. Some microbes are pathogen only for human. That's why we cannot introduce the pathogen, the dangerous pathogen, to human. So, as an example, in HIV viruses, which is pathogen only for human, we cannot prove the third postulate of Koch because it is unethical. The fourth exception is that the disease do not appear until many years after a specific infection. Again, the example of this exception is also the HIV, because we cannot prove the third and the fourth postulate in a short period. We have to wait until the disease is appear. Due to those exceptions, then we have to revise some of the Koch postulate, and these are the guidelines to establish the cause of infectious disease. For the first Koch postulate, the microbe must be present in every case of the disease. For the molecular Koch postulate, the phenotype or property under investigation should be significantly associated with pathogenic strains of a species and not with non-pathogenic strains. And molecular guidelines for establishing microbial disease causation, the nucleic acid sequence of a putative pathogen should be present in most cases of an infectious disease. For the second postulate, the microbe must be isolated from the disease host and grown in pure culture. Specific inactivation of the gene or genes associated with the suspected virulence trait should lead to a measurable decrease in pathogenicity or virulence. And the molecular guidelines for that is that the nucleic acid sequence of a putative pathogen should be absent from most healthy controls. If the sequence is detected in healthy control, it should be present with a lower prevalence as compared with the patient with disease and in lower copy number. For the third postulate, the disease must be reproduced when a pure culture is introduced into a non-disease susceptible host. The molecular Koch postulate is that reversion or replacement of the mutated gene with the wild-type gene should lead to restoration of pathogenicity or virulence. And the molecular guidelines for that is that the copy number of a pathogen-associated nucleic acid sequence should decrease or become undetectable with resolution of the disease and should increase with relapse or recurrence of disease. The fourth postulate: the microbe must be recoverable from an experimentally infected host. And there are 3 molecular guidelines for that. They are: first, the presence of pathogen-associated nucleic acid sequence in healthy subjects should help predict the subsequent development disease. Diseased tissue should have higher copy number than normal tissue. And the last one is that this sequence-based findings should be reproducible. When we talk about the infectious process, then we talk about the entry of the microbe into the human being, into the body, and then colonization of the microbes, adhesion, and invasion into the cells, and then the pathogenic action. In the first slide, we have talked about the 3 main components of the disease. They are host, agent, and environment. This slide shows you how the correlation between the microbe or the agent and the host. There are 3 components of the microbe which influence whether the microbes will produce the disease or not. First is the virulence factor, the percentage of optimal infectious dose, and the correct portal of entry. The virulence, of course, should be high. The percentage of the infectious dose should be optimal, and to produce the disease, then the microbes should enter the body through the correct portal. And from the host, there are 3 components as well. They are the genetic profile that resists the microbe. This is the component of the non-specific defenses, and then previous exposure to microbe. This is the specific immunity, and the third one is the general level of health, like the nutrition, and is there any underlying disease or other things. When there is a previous exposure to the microbe, means that the host has already had the antibody to the microbe. When there is no previous exposure, means that the host doesn't have any antibody to that particular microbe. Considering these 2 main components, there are 3 possible outcomes. So when, for example, when the microbe has high virulence, optimal infectious dose, but it entry through the incorrect portal, then it will not produce any disease, and the microbe will pass through unnoticed, and it will be excreted from the body. The second outcome is the microbe become established without disease. This outcome can be appeared when the microbe has the virulency, enter the body through the right portal with the optimal dose, but the host has already had the specific immunity to this microbe, then the disease will not be produced. The third outcome is the microbe causes disease, and this will be appeared when the microbe has the virulence and enter the body through the correct portal with the optimum dose, and the host has not had the antibody yet. There are three main portals of entry: skin, mucous membrane, and placenta. As long as our skin is intact, then there is no microbes will enter the body. But if there is a microlesion, then it is a very good entry of the microbes to enter the body and produce diseases. The second portal of entry is mucous membrane. We know that all of the tracts in our body are surfaced by the mucous membrane, and conjunctiva is also the thin membrane covering the surface of the eyeball and the underside of each eyelid. As long as the mucous membrane is intact, it is also no microbes enter the body. When there is a microlesion, the microbe will enter through this. The third portal of entry is placenta. There is a barrier, but some of pathogen can break the barrier and enter the fetus to produce diseases. There is a parenteral route. It is actually not a portal entry, but instead a means by which the portals of entry can be circumvented. Once the microbes enter the body, they will try to colonize the surface of the body. And to colonize a site populated by normal flora, it is of course not an easy thing. The new arrival must compete successfully with established organisms for space and nutrients, overcome their toxic product, and counter the body's defenses aimed at protecting the surfaces, which is IgA proteases. When the microbes successfully colonize the surface of the body, then they will attach themselves to cells. In this process, they need the adhesion factors. There are some adhesion factors we know. First is the specialized structures, which is belongs to protozoa. We call it adhesion disc. There is also suckers or hooks in helminths, and also ligands. Ligands is the surface lipoprotein and glycoprotein, and we can found ligands in bacteria or viruses. The ligand in bacteria is named adhesion, found on fimbriae, flagella, and glycocalyces. The second adhesion factors is ligands, which can be found in bacteria and viruses. In bacteria, we call it as adhesin, and in viruses, we call it as attachment protein. There are two types of bacterial adherence mechanisms. They are pili. This is the rod-shaped protein structure that extend from the bacterial surface and bind to host cell surface molecules. The second one is the afimbrial adhesins. This is the bacterial surface protein that are not organized in a rod-like structure and mediate tight binding between bacteria and hosts. There are some factors support the adherence mechanism. Pili and fimbriae will mediate the attachment, and the pili constantly loss and reform, and it allows some bacteria avoid host defenses. Adhesion protein and also signal transduction. Loss of ability to make ligands happen because of genetic change or mutation, or exposure to certain physical or chemical agent, and the loss will make the microbes harmless or avirulent. Some bacterial pathogens do not attach to host cells directly, but instead interact with each other to form a sticky web of bacteria and polysaccharides called a biofilm. Where do the microbes attach to the host cells? We know that there are some receptor molecule on host cells, and this receptor molecule typically are glycoprotein containing sugar molecule such as mannose and galactose. This receptor molecule is present not for the benefit of the infectious agents, but they have specific functions in the life of the cells. The receptor molecule present only in certain cells, which are then uniquely susceptible to infection, and also specific for certain pathogens. For example, the Neisseria gonorrhoeae has adhesion on its fimbriae that adhere to cells lining the urethra and vagina of human. When the disease is produced, there are stages that can happen. First is the incubation period. This is the time started from the exposure of the infectious agent until the appearance of the first symptoms. There is no sign or symptoms in the incubation period. After that, there is a prodromal period with very unspecific symptoms, and then the illness with the specific symptoms and more severe symptoms, and after that there is declining, the symptoms are declining, and then convalescence, when there is no sign or symptoms appear. During all those periods, the microbes can be excreted from the body through the portal exit. When it is excreted, it can be transmitted to other human beings, and there are some transmission mode as we see in this slide. Some microbes can be transmitted directly to other human beings, but also can be transmitted through vectors or reservoir. The vector is, for example, mosquito, and reservoir are mostly animals which may cause the zoonotic diseases. This slide shows you the microorganisms or the pathogens which can be transmitted through milk from human or cow. The same process occurred in the pathogenesis of viral infection. The virus should enter the host, contact and enter susceptible cells, replicate within the cells, release from the host cells, and then there is interaction with the host immune response, and this interaction will produce three outcomes: cleared from the body of the host, establish a persistent infection, or even kill the host. And after that, the microbes or the virus will be shed back into the environment. This slide tells you how the virus enters the body and then attach to the host cells. Adsorption or attachment is a process of penetrates of host cell to gain access to the cell's replicative machinery, and this will happen if the virus have a specific protein ligand. There are two kinds of viruses: enveloped virus and naked virus. The enveloped virus will use spikers to attach to host cells. The spikes is the viral protein that protrude from their membrane. And the ligands of the naked virus is part of their capsid protein. As I told you before, that the interaction between virus and the immune cells will produce three kind condition. The first one is if the immune system, especially in this case is the cellular immune response, successfully kill the virus, then the entry of the virus will not produce any disease. As we know that virus is an intracellular microorganism, so it must be in the cell. When the response immune works successfully through the cellular immune response or the natural killer cells, this process will kill or will cause the cell death, and it causes also the death of the viruses live in the cells. but if the response immune failed to kill the infected cell, and neither T cell nor NK cell can recognize virus infected cell expressing fake MHC class 1 protein, then the cell will survive and also the virus will survive. Binding of a virus to its receptor on the host cell surface result in penetration of the cell or the delivery of virus nucleic acid to the cytoplasm of the cells. To be able to produce a disease, the virus must enter the cells, and there are three ways how they enter the cells. First is they directly enter the cells. The second one, the fusion of the viral envelope with the cell membrane, and then subsequent uncoating. And the third one is the endocytosis and then release of the nucleic acid from the capsid or uncoating. After the entry the cells, it will replicate, and after replication they will release from the host cell, and there are two mechanisms of release. First is very dramatic and result in relatively large numbers of virions leaving the host cell at the same time and host cell death, and the second one is through budding or blabbing. There are two kinds of virus-host interaction. First is the cytopathic viruses, and the second one is the non-cytopathic viruses. Cytopathic viruses will ultimately kill the host cell, and the result is often local necrosis. And non-cytopathic viruses will not immediately produce cell death and result in latent or persistent infection. There are some viruses known to be the cause of cancer. In the normal condition, division of cells in a mature multicellular animal is under strict genetic control. And in the neoplasm, the phenomenon is uncontrolled cell division. The cells undergoing neoplasia are said to be neoplastic, and a mass of neoplastic cell is a tumor. And there are two kinds of tumor, benign and malignant. This slide tells you about the theory of the introduction of cancer in human. The theory suggests that more than one hit to the DNA, that is, any change or mutation, whether caused by a virus, is shown here in the second part of the figure. The yellow one is the insertion of the virus, or various physical or chemical agents is required to induce cancer. Viruses cause 20 until 25% of human cancer in several ways. They carry copies of oncogenes as part of their genome, promote oncogenes already present in the host, or interfere with normal tumor repression when they insert into repressor genes. So in summary, infectious diseases are produced through the imbalance of hosts, agents, and environment. There are some factors of hosts and some factors of agents that contributes to the production of the disease. That's all about the basic concept of infection, and I hope you will learn more by yourself. And if there is something that you don't understand, you may contact me or we can discuss it together.